is here. I'd like to introduce Shirley Hui, who is will present for the next hour about her thesis project to computationally predict PDZ domain-mediated protein interactions. Shirley was an undergraduate at Waterloo in computer science, and then she did a master's in bioinformatics at Waterloo with Forbes Burkowski's lab, and that was on 3D structural alignment using dimensionality reduction techniques. And then she was also a software developer at a medical imaging company. So she gained some great experience there as well. And now, and for the past, since I guess 2007, she's been here in the molecular genetics department at the UT, working on this thesis project. And thank you for looking forward to hearing the talk.
Okay. Thank you. So today I'm going to talk to you about my thesis project, which is the computational prediction of PDZ domain-mediated interactions. So we all know that proteins interact with each other in order to facilitate a variety of biological processes. So if we're able to detect how these proteins interact with each other, then we can construct protein interaction networks. And this can provide insight in how these biological processes are organized. So there are different ways to detect protein interactions. There are experimental methods. There are in vivo methods which can detect interactions within the native environment of the cell. There are in vitro methods which can detect complexes or be used in a high throughput manner. However, when there are many thousands of proteins involved, as in the case of a typical proteome, in vivo, small scale in vivo methods don't necessarily scale up well, and high throughput experiments often face high false positive rates. There are also computational methods that have been developed, and these can help lend additional support to experimental findings or to help biologists prioritize experiments. They can be briefly categorized into physics-based or statistics-based methods. And these methods basically involve taking information about proteins and then applying algorithms in order to predict if proteins interact or not. However, many of these methods face challenges. So, for instance, physics-based methods often require three-dimensional structures of the proteins to be available. Some statistic-based methods are rather simple and only can consider short sequences. And so the problem of actually predicting protein-protein interactions is very difficult and is not adequately addressed by any method currently. However, if we were able to accurately predict protein interactions, then we could do something like scan a given proteome for interactions and then build a comprehensive protein interaction network in an application I call proteome scanning. So, although the computational prediction of protein-protein interactions is very difficult, many protein interactions are actually mediated by peptide recognition domains, which we know a lot about, and many have been studied. These domains are evolutionarily conserved. They can fold independently and are often found combined in different ways to form larger proteins, especially signaling proteins. So they're often involved in eukaryotic signaling systems. And so some of these signaling proteins have important roles such as photoreceptor or cell growth signaling, or some of these proteins are found in important pathways such as the Hippo pathway. But one of the advantageous things about these protein domains is that they bind their targets through the recognition of simple linear motifs. So the computational prediction of this type of interaction is much easier than trying to computationally predict protein-protein interactions in general. So for my thesis, I'm focusing on the PDZ domain. The PDZ domain consists of 80 to 90 amino acid residues which fold into this type of structure that consists of 5 to 6 beta strands and 2 alpha helices. They bind hydrophobic C termini, which bind into a hydrophobic groove formed between this beta strand and this alpha helix. The binding specificity of the PDZ domain has been well studied, and the specificity has been broadly categorized into 2 classes. The first class prefer a serine or threonine at the 2 positions from the C termini, and a valine or leucine at the last position, whereas a class 2 specificity prefers hydrophobic residues at these 2 positions. PDZ domains also have interesting biological roles. They're found in proteins to help establish and maintain cell polarity, localize protein complexes to specific areas of the cell, or to facilitate efficient photoreceptor and neural signaling. So my thesis focuses on the computational prediction of PDZ domain peptide interactions. So in order to build such a predictor, I used a framework that I followed for all the projects within my thesis. And so for the framework, it consists of 4 major components. The first component involves collecting data. So the types of data that I collected were interaction data from experiments or curated sources, or computationally generating interaction data. The other type of data I collected was structured data for PDZ domains, and these were experimentally determined or homology modeled. And then the second component is feature encoding, and this involves taking the interaction or structured data and then encoding them into numerical format to allow for the predictor to learn patterns from it. And 2 types of encoding methods or information that I encoded are the sequence-based and structure-based information. The third component is actually to train the predictor. So I used a support vector machine, which is a binary classifier. It basically takes positive information and negative information and tries to find a line that best separates the 2 classes from each other. Predictions are made by determining which side a given data point falls. So if it falls on this side, then it's predicted to be positive, otherwise negative. So this is a simplification of the actual problem because PDZ domains don't just bind yes or no. There's actually a strength associated with their binding. However, for our purposes and because the interaction data is usually binary, we are solving the binary form of the problem. So the fourth component is to evaluate the predictor, and there are different strategies that I used. The first is to estimate the performance of the predictor using cross-validation, which involves holding out a subset of the data for testing and then training on the rest of the data. And the other type of testing that I did was blind testing, which involves utilizing data that has not been used for training before and then gaining an unbiased assessment using that type of data. And then the actual measures that I use to score or to assess the actual predictor's performance is the AUC, or area under the curve. And there's 2 curves that I construct in order to assess predictor performance. The first one is an ROC curve, which measures the trade-off between true positive rate versus false positive rate as the predictor's discrimination threshold is varied. And so this value, this AUC score, the area under this ROC curve, ranges between a value representing random to 1, where 1 represents a perfect predictor. Higher values closer to 1 or better. And the second type of curve I construct is a precision recall curve, which measures the precision versus the recall of the predictor. And again, the area under this curve ranges between a value representing random performance up to 1, which represents perfect performance. So AUC measures closer to 1 indicate better predictor performance. So an overview of my thesis is that I'll first talk about a sequence-based predictor that I developed. This predictor only utilizes sequence information about domains and peptides. And then I'll talk about the second project I worked on, which involves utilizing domain structure information and peptide sequence information to build a predictor. And then the final project involved taking the predictions from these two predictors and then filtering them using an integration system to filter for physiologically relevant and high confidence interactions. These interactions I then used to build a PDZ-mediated protein-protein interaction network in human. So for the first project, the predictor that I built takes a domain sequence and a peptide sequence and then predicts whether or not this represents an interaction. The data that I used to train the predictor are from 2 high throughput experiments. The first one is a phage display experiment in human. This involved using random peptides displayed on the phage surface and to add them to domains which were bound or immobilized. Any bound peptides were then washed away, and any that were bound were amplified and then added back to the domains, and this process was repeated multiple times. And in the end, approximately 2,000 positives were identified. The second experiment involved a protein microarray experiment in mouse, and this experiment utilized approximately 200 fluorescently labeled genomic peptides, which were used to probe approximately 160 PDZ domains on protein microarrays. The array positives were then identified and then retested using fluorescence polarization. Binding affinities were then calculated, and then depending on whether the affinity exceeded a certain threshold, they were identified as positive. So there were approximately 640 positives identified and 1,300 negatives. So there were a few things that we had to do before we could use the phage display data for training. It first had to be pre-processed. The first thing we had to do was to filter out non-genomic interactions, and we found that this was necessary, especially because we're interested in proteome scanning, and so we wanted to enrich for genomic interactions in this training set. So we removed any interactions that involved peptides that were not seen in the proteome. And the second thing that we had to do was generate additional artificial negatives, because this data set only consists of positive interactions, and we need both positive and negative interactions to train the support vector machine. And this resulted in approximately 1,000 positive interactions and 2,000 negative interactions. These interactions then underwent a sequence-based feature encoding, in which we encoded sequence information about domain peptide residue pairs at 38 contacting positions in the given domain and peptide. And these 38 contacting positions were derived from a single domain peptide complex structure. So for each contacting position, there's a binary matrix which encodes the specific amino acid pair. Using different cross-validation strategies. These cross-validation strategies involved holding out subsets of the training set, and the performance measures are all close to 1. And so this indicates that the predictor, well, we estimate the predictors should be able to perform well in practice. One thing we did notice was that the strategies that involve holding out subsets of domains had slightly lower performance scores than the other strategies. And so this suggested that maybe the predictor performance depends on how similar the domains are, or the testing domains are, to the ones in the training set. So to actually determine the amount of dependence, we ran a leave one out domain cross-validation strategy, which involves holding out single domains and their interactions for testing, and then training with the rest of the data. And then we categorized, or we grouped the performance measures based on how similar the held out domain was to the domains in the training set, and found that if the domain was very similar, then the performance was high. Compared to domains that were less similar, the performance was lower. So there is a dependence on sequence similarity. And we also performed the same analysis using a naive nearest neighbor predictor, which simply uses sequence similarity as its criteria for prediction. And compared to this naive predictor, the sequence-based predictor does perform better, except when domains are less than 60% similar, in which case the performance are comparable.
So sure, this is sequence, these are the domain contacting residues, or this is the entire domain sequence?
This is the binding site. Those, yeah, sequences in the binding site. Okay, so blind testing was also performed using genomic interactions from mouse, worm, and fly protein microarray experiments. And for worm and fly, the performance is pretty good. However, for the mouse test, the performance is much lower. So to investigate why this was the case, we computed the average domain sequence similarity in these test sets to the ones in the training set, and found that the mouse domains were much less similar than the ones in the worm and the fly test. So this lower performance is not unexpected. However, in general, this shows that our predictor is able to correctly predict many interactions of different organisms. We then performed proteome scanning for human, fly, and worm PDZ domains. And we validated a subset of the interactions for a number of PDZ domains. For the human domains, the average true positive rate is approximately 65%, with lower rates for fly and worm. However, the false positive rates are very low, approximately 4%. So these results show that the predictor is able to correctly predict many known interactions at the expense of very few false positives. And because validation data is limited, we also compared the predicted binding specificities, which I visualize as sequence logos here, versus the experimental binding specificity obtained with phage display experiments, and the average similarity was approximately 67%. Finally, we compared the sequence-based predictor. We compared the predictor to other predictors that are available for use. 2 of them are state-of-the-art, and 1 of them is a baseline predictor. We compared the F1 measure, which is a single value or single score composed of precision and recall, and we found that the predictor has a higher F1 measure for different organisms compared to the other predictors that we tested. And as well, the false positive rate is also lower compared to these other predictors. So compared to other predictors that are available, our sequence-based predictor is better at the application of proteome scanning. Okay, so since a sequence-based predictor we found depends to an extent on domain sequence similarity, the second project involved building a predictor that utilized domain structure information, and it takes as input a given domain structure and a peptide sequence, and then predicts whether or not this represents an interaction. So for this project, we used 83 structures for training. A number of them are from the PDB and our X-ray or NMR structures, and the rest of them were homology modeled. So for each structure, we extracted information that is important for protein folding and stability, and these features pertain to 10 core binding sites in the domain. These 10 core binding sites were defined by 9 different PDZ domain peptide complex structures. And the information that we extract or encode for these different positions include information such as solvent accessibility, patterns of hydrogen bonding, and electrostatics, etc. And then the interaction data that we use for training is actually the same as for Project 1, except for when we were initially training the predictor, we noticed that it had problems. It specifically for certain domains, or for many domains, it would predict many true positives. And so this problem we termed overprediction, and we eventually discovered that this probably had to do with the fact that the negative interactions that we were using didn't adequately, or don't adequately represent all types of negatives. And so the predictor was having problems with this overprediction. And so we generated additional artificial negatives for training using a semi-supervised learning method. And in the end, we ended up with 940 positives and 1,800 negatives. The peptides in the interaction data set were then encoded where a given peptide, for a given peptide, each residue is encoded using a binary vector where there are all zeros in the vector except for one, indicating that residue that's being encoded. So we also performed cross-validation to estimate the predictor's performance. We used the same strategies as we did in the first project, and all the performance measure scores are close to 1. However, again, we noticed that the strategies involving holding out sets of domains were slightly lower than the other strategies. So again, we determined whether or not there was a dependence on sequence similarity for our structure-based predictor. And so what we did was we performed a leave domain out cross-validation. And in this strategy, we pulled out subsets of domains, but for the training set, we only retain domains that are lower than a specified cutoff. And so the performance measures were grouped based on the cutoffs that we use. And again, for domains that are very similar, the performance is better than domains that are less similar. We also performed the same analysis using the sequence-based predictor from the first project. And when we compare the performance, if the domain is greater than 70% similar, then the performance is comparable. But if the similarity is less than 70%, then the structure-based predictor is better. Although both predictors still depend on domain sequence similarity. Again, we performed proteome scanning for human flying worm PDZ domains and validated a subset of these interactions that we predicted. The human proteome scanning, we are able to predict, or the average true positive rate is approximately 80%, with lower rates for flying worm. The false positive rates are also very low. So again, we're able to correctly predict many known interactions at the expense of very few false positives. And again, if we compare experimentally determined and predicted binding specificities, the average similarity is approximately 68%. And then some specific examples of if we compare sequence-based and structure-based binding specificity, we can see that in some cases, for instance, this PDZ domain, the structure-based predictor predicts a hydrophilic threonine at the -2 position, which is documented in the literature, whereas a sequence-based predictor predicts a hydrophobic isoleucine residue at that position. For this PDZ domain, in the literature we find that it is bispecific for tyrosine or tryptophan at -1, which the structure-based predictor does detect. However, the sequence-based predictor doesn't predict a the preference for tryptophan. And then finally, another example, for this domain, hydrophilic lysine at -2 is documented. However, there's no such preference for this residue at the -2 for the sequence-based binding specificity. So there's examples where predicted binding specificities are supported by published results. Okay, so the next thing I did was to compare the biological functional enrichments between structure-based and sequence-based targets. So that's what I did, and I created an enrichment map which compares the enrichments between these two sets of predictions. In this map, which is a network, the nodes represent enriched terms. The nodes consist of borders, which represent structure-based enrichments, and the centers represent sequence-based enrichments. The borders and nodes are colored according to a gradient, with red indicating a high enrichment level, and lighter color indicating a low enrichment level, and gray representing no enrichment at all. Edges in this network connect nodes that are related, and then I just circled clusters of nodes that were related and gave them more general labels. So from this map, there are biological functions that are enriched, and we know that they involve PDZ domains. So for instance, neurodevelopment, cell adhesion, ion transport. However, there are examples where there are processes which are enriched only in structure-based targets, such as neuron projection morphogenesis, innate immune response signaling, and also there are functions that are enriched only in sequence-based targets, such as active movement, nuclear transport, and membrane fusion. So this map makes it easy to identify similarities and differences in biological function enrichment between structure-based and sequence-based targets. And in fact, these predictors are different, however they're complementary. In particular, the structure-based predictions result in a 40% increase in the number of PDZ-mediated protein-protein interactions that can be predicted. So it highlights the importance of using different features in order to increase the coverage of what we can currently predict. So because our predictions are based on sequence or structure information, the interactions that we're predicting are not necessarily physiologically relevant. And so in order to identify such interactions, I used a Bayesian integration system, which was developed by Shirley Jane in our lab, and then took the filtered interactions and then built a PDZ-mediated protein-protein interaction network in human. It takes information from different biological evidences, and then calculates a probability of whether or not this interaction consisting of protein A and protein B is physiologically relevant or not. And there's different features that we use. So for instance, we compute the average gene expression correlation between protein A and protein B, or we determine the amount of similarity in biological process or molecular function or cellular location of the two proteins, or we calculate whether or not a given domain in protein A and a given domain in protein B co-occur, or we calculate the maximum conservation of the protein tails according to various model organisms. And so these features are integrated by computing the probability of seeing these features in a positive gold interaction set or a negative gold interaction set. And the probability of so if the probability of seeing these features in the positive set is greater than in the negative set, then protein A and protein B are predicted to be physiologically relevant. So Chobit performed a tenfold cross-validation to estimate the performance of this integration system, and the AUC scores were both very good. And he also performed another type of cross-validation to determine the contribution of the individual evidences by training individual predictors that were trained only using one evidence source. And the results show that using individual evidence sources by themselves results in inferior predictor performance. It's only when you use all of the evidence sources together do you achieve the best performance. So using the integration system, the majority of our predictions are actually filtered out. Specifically, 90% of our interactions are filtered out. So ideally, if our system is working correctly, then the number of false positives that are filtered out should be greater than the number of true positives being filtered out. And so we validated our interactions using known interactions from databases and the literature. And so we found that there were indeed more false positives filtered out than true positives. I then took the interactions and then built a protein-protein interaction network mediated by PDZ domains. And so this network is different from the ordinary protein-protein interaction network in that there's two types of nodes in this network. The pink ones represent PDZ domains, and the light purple ones represent PDZ targets, or the C termini. And then these nodes are connected by approximately 3,400 edges, or approximately 3,400 interactions are represented in this network. So using this network, we can visually identify candidates for further study of specific properties of PDZ domains. So for instance, looking at the different clusters in this network, we can identify promiscuous versus selective PDZ domains. So promiscuous domains would be, for instance, Shank3 PDZ1, which is predicted to bind over 200 different targets. These targets are enriched in different biological processes, such as neural development, actin filament organization, compared to more selective PDZ domains, like RGAP21 PDZ1, which has 5 binding partners, and they are all enriched in GTPa's activity function. Another property of PDZ domains that we can study is cross-selectivity, which refers to the fact that PDZ domains often bind the same or similar sets of targets. And this can be measured, the amount of cross-selectivity can be measured using an overlap score, which simply computes the percentage of targets that are similar between two PDZ domains, one representing complete overlap, and 0 representing no overlap or very low cross-selectivity. So an example of domains that have high cross-selectivity are ones that are related or paralogs. So PDZ3 of the disk large 1 gene of family of genes is one such example, has a very high overlap of 0.6, and this is reflected in its binding specificity, which is also very high. And so this is not unexpected because they are related, they have a very high binding site similarities, which is probably the reason that they bind similar targets. However, there are examples where there are PDZ domains that are not related, and they have a reasonable amount of overlap, and their binding specificities are very similar. And so, the actual binding sites are very different sequence-wise. So this might, the fact that they bind similar targets could be related to the fact that structurally their binding sites are very similar. On the other hand, there are examples where cross-selectivity is minimized, so the overlap is very low. And one example is the MPDZ protein, which has many PDZ domains along its protein. And the binding specificity similarity is very low, so this indicates that the domains each bind very different targets. And so this is probably important for this protein because it's a scaffolding protein, and so this allows the domains to bind different targets simultaneously during protein complex assembly. I also created a map, another map, to compare functional enrichments between our predicted physiologically relevant interactions versus one's interactions that are known in a database called IREF index. And these interactions are protein-protein interactions that involve PDZ domains. So compared to the previous map I showed, there are many processes that are enriched also in the current map, including white blood cell differentiation, innate immune signaling. However, there are some examples of functions that are no longer present in the current map, and these are likely due to the fact that they're low confidence or not predicted to be physiologically relevant. And then there's a set of biological functions that are enriched or have targets that are novel, so they're not found in the database or in literature, and they could be followed up in further studies. So finally, we performed membrane yeast twohybrid to validate a number of our predictions. And in this experiment, it involves splitting a ubiquitin molecule into two fragments. One is attached to the bait, one is attached to the prey and transcription factor. And if there's no interaction, then these two units do not come together, and then there's no activation of these reporter genes. However, if there is an interaction between bait and prey, then a fully formed ubiquitin molecule is created, recognized by enzymes which cut off the transcription factor, which then enters the nucleus to activate the expression of these three reporter genes. So these experiments were carried out by Zhang Yao in the Stagler lab, and we selected 5 predicted interactions involving the frizzled 7 protein, which is a G protein coupled receptor. And we picked these interactions because they were novel, not found in any databases or in the literature. And so the bait and the prey were cloned and then co-transformed into yeast cells, and these yeast cells were grown in selected media to determine interactions. So on the left-hand side is interaction selection using 2 of the reporter genes. And in this case, if there is an interaction, then we will see colonies growing. And then in this panel, this tests for interactions using 3 reporters, and if there is an interaction, then blue colonies will form. So for our purposes, we determined a prediction to be confirmed if there was reporter expression in either of these cases. So for 4 of these cases, 4 of these predicted interactions were positively confirmed. And so these proteins are actually known to be involved in cell polarity or proper cell junction formation. So this suggests a possible role for PDZ domains in terms of assembling protein complexes in these processes. So in summary, I've talked about a sequence-based predictor that I developed and a structure-based predictor that I developed. And these predictors are now available online for people to use and support predictions for the majority of human, mouse, worm, and fly PDZ domains. And then I also talked about a third project in which I took these predictions, put them into an integration system, and then used the resulting interactions to build a physiologically relevant PDZ-mediated protein-protein interaction network in human. So some of the things that we can do with our current network include training the predictor with additional features, such as information about domain flexibility or binding site shape, and this would increase the coverage of our current PDZ interaction network. Another thing we could do is we could build predictors for other peptide recognition domains, such as SH3WW and other domains, and then perform proteome scanning. And this would result in the construction of a high-resolution, comprehensive peptide recognition domain-mediated interaction network. And then finally, we could use the network to also map changes from evolutionary or disease-related mutations, and this would result in identifying interactions that are abolished or new interactions that are added due to these mutations. So that's the end of my talk, and I'd like to acknowledge all members of the Baderlab, especially those who have helped over the years in different areas of the different projects I've worked on. I'd also like to thank my committee members for their insights over the years, and I'd also like to thank members in different labs that performed experiments to generate data for me to train the predictor as well as to validate the predictor.
The department asks that only the attendees that are not on the exam committee ask questions, and then because the exam committee has a couple of hours time to ask questions. So, any questions?
Shirley, you mentioned some domains that had similar binding specificities, even though you said they're not related, but all PDZ domains are related. But do you have any idea whether these constitute convergent evolution or traces of similar specificity of an ancestral domain that goes very far back in time?
So that one example that I showed.
One was MAS2, I didn't complete.
Yeah, I think, well, I haven't actually looked into it in much detail. It could be convergent evolution, although I have to look into it more. But yeah, so
But in principle, you have the tools now that you're able to go back in time, construct ancestral sequences, and try to pursue in a diachronic fashion how the specificities evolve. Is that correct?
I'm sure you could definitely do something like that. So
Cool.
something that we could do in the future, I suppose. Yeah.
Any other questions? I should also mention that Shirley did actually do some of the experiments relating to the yeast hybrid data with the help of Andrea, who's here. So she did computational stuff and also got her hands wet a bit. So if that's it for questions, let's thank Shirley one more time. It's been a pleasure having you in the lab, and obviously you're around for a bit more. So that's it. Thank you very much.
Sometimes you need to twist it. Yeah.
It might not be that pressurized. We don't have a saber.
Wow, this is tight.
Almost. Shake it a bit. Yay! So we have so that's sparkling grape juice, the peach juice.
Oh, okay, great, perfect.
And then this is actual champagne. So we want it, if you want it, actual champagne.
Okay, do you want to?
I'll probably have a beer. Does anyone want anything?